So again, on Thursday, this Thursday, 2 days from now, there will not be a lecture. And there will also not be a lecture on the 10th, which happens to be the Tuesday in a week from now because of the student assembly. And then the next one that will also not happen is the 3rd of December. The special lecture from Jana Schmidt is now fixed. Oh, it's not on here. To some point, I'm not entirely sure, I believe 15th, December 15th. I will show you that next time. I want to show a few people here. That's Michael Lunik. I like him. But Yanayo Fran, so some of the slides in the introductory come from Yanayo Fran, who's now at Bailan University. Runs a lab there and sits in Boston. That's another beauty of science, by the way. Marco Punta, currently in Paris. Sina Reyes, and Avner Schlesinger here, who is now, I will show another slide, back in Mount Sinai in New York. Kaas, now Brzezinski, who is now doing his PhD here in the New York Genome Center. Okay, genes and numbers. So the common thing in life are the genes. Somehow the idea is that genes explain, in the olden days of Leonardo da Vinci, the idea was that sort of shapes explain how life fits into the universe. Now the idea is that genes explain how it fits. What did life begin with? So I told you about this sort of dogma that says there's the information flow from RNA to DNA, from DNA to RNA to proteins. Proteins are the machinery of life. How did life begin? Has anybody ever heard any model? What's the idea? Are you asking me a question, or are you telling me an answer? I mean, I'm not. The word proteins. Yes, I heard proteins. And the life begins with proteins. Is that what you say?
DNA, not with RNA, but with proteins.
That's what you say.
Yeah.
Anybody? Any comment on that?
As far as I read, it's the current assumption is it starts with RNA, and RNA needed proteins to proliferate.
Exactly. So again, this is a hypothesis. That hypothesis got Ig Nobel Prize already 40 years ago by now. And the experiments that Ig has done sort of showed something in principle. It's still not overall, I'm sort of wrapping up 40 years of research here in a very negative statement. I believe we have not advanced much beyond the model. So this is a model that everybody somehow accepts, but the proof is missing. But the idea really is life starts with RNA because RNA are small molecules that can self-replicate. RNA have issues with the types of functions they have. And an argument that comes here, ultimately one group is Andrei Lupas here from the Max Planck Institute in Tübingen, who argues, well, RNA needs, as Dimitri correctly says, RNA needs slaves to do some enzymatic function, proteins. In fact, it starts with peptides, so short strings of amino acids. And he argues the types of amino acid, I told you that there are 20 different amino acids, and I will show you again today a few of these numbers. The types that have been chosen because they are the ones that are good slaves for RNA. Okay, so they do exactly the type of functions that RNA need to do in order to survive if life started with RNA. And self-replication is one of the important functions of life starting. So you have a molecule that self-replicates, and part of that process is done by proteins. So Andrei Lupas tries over his lifetime to, in fact, if that were true, he argues, then we would have some sort of marker or some history built into proteins. So if they need to help RNA, they need to bind RNA. If they need to bind RNA, some old fragments of these proteins, some should show that they are capable of binding RNA. And we should find them in a lot of modern proteins. And he has been tracing that, and for certainly ballpark of 50 proteins, he has a very good case now. A lot of those with a lot of experimental information. So it's a lifetime work. And here's just the evolution of the rift monomer, so one particular type of these. There are others. Now, if that were the explanation, So if RNA really started that way, RNA is sensitive to an environment, so it could be easily degraded. So what do you need next? Exactly. You need to essentially put something around it here, a lipid bilayer. Here shown as in a simple cell, how densely packed are cells. So if that sort of were the membrane, and the membrane would be like a bag, and there's all kinds of stuff in this bag, in a typical cell. How do you imagine this cell to be? How densely packed is it? How much material is in there? If this would not be a lipid bilayer, but it would be a plastic bag, a shopping bag, how much would you have to put into it to have a model for the cell? 30. And what material would you put in?
30, 30 full of stuff and 70 free.
So 70 free. So that means you would have such a bag that sort of wobbles around. Or would you fill it with something else?
Something else.
What is the something else?
Water.
Okay, so you believe it's 70 water and 30% some dense material. This is in fact very close to what I've been having as my image in my head. It turns out this is the wrong way of thinking about it. It is exactly what we need in order to imagine how proteins fold and all of that. The last semester was about protein structure formation. It was always folding in liquids, because proteins are solved in liquids, in water. They're surrounded by water. But it turns out that the water they're surrounded by is very little. So it's actually better to imagine this as a bag filled. Yes, there's water in there, but filled with, I don't know, hard grains that sort of are surrounded by water. They're all liquid, but the whole thing is like a mesh that is really condensed. It's a solid, more like a solid state than like a liquid in which things swim around. And this is just one image here from David Gutzel, who makes these beautiful, and this I believe is another reason this is the smallest bacterium, Mycoplasma genitalium, and what he has dedicated his life to is essentially make drawings of biology. You cannot make pictures of these, and if you made photos, so the cell you see on a microscope, but you don't see at that resolution. And his skill really is to describe biology by drawings in a way that are really amazing. And they are not imagined, so it's not the artist who is imagining things, it's a science-driven new discipline, if you want. Again, DNA, RNA, proteins, and the numbers we went through, so you would have about 20,000 different proteins. Now, the common assumption is that proteins adopt a unique three-dimensional structure, and they do their function through that unique three-dimensional structure. What is a protein? Essentially, it's a string of amino acids. One particular protein is shown here. The name is here. It's from this organism, and that is essentially the 20 amino acid letter code written in one line. And the idea really is, and Christian Anfinsen got a Nobel Prize for the groundbreaking experiments in this direction, that in principle the chain thrown into a solvent, water, and we're back to the bag of water idea, adopts a unique three-dimensional structure. The unique three-dimensional structure is responsible for a unique function. There's a whole library of folds of different shapes that do, yeah?
Maybe it's a very silly question, but what was the shape of the protein before it was thrown into the water?
So, in principle, so first of all, first statement, we do not know, because we cannot look at it. We can only look at it. So the way we can determine the three-dimensional structures here, the most dominant way by which we know these images is through X-ray crystallography. For that, you have to grow a crystal of the protein. It means you have to get it in high density, and you shoot X-ray beams into it, and from the diffraction pattern, you find out where the atoms were. Okay? Now, this is not a protein in its natural environment. So it's a different protein already. But the way this crystal works is because you have the same in many crystal units. So you have the same protein in the same shape over and over again, and there is a consistent scattering because you have a repetition of the same. The moment you have a region here in this protein, where in one part of the crystal I don't know, you have the helix like this, in another part you have it like this, then the scatter pattern is very confusing. It's not consistent. If the protein is a stretched out chain and wobbles around, you would get exactly that. You would get a scattering pattern that's absolutely inconsistent. That means you wouldn't see anything. And under the microscope, you can't see it. So what we can see is that proteins don't give a consistent pattern. Now, does it mean that they mostly do sort of like a string in water? Or does it mean something else? We don't know. We do, however, believe that molecular dynamics, and I'll get to that in a moment, for that problem, for that question, is a good answer. And molecular dynamics would say it's just like a string in water. Or string is not the right word. Something that is immersed in the water, but swims and doesn't sink, swims around and has flexibility, and waves come. Yes?
And another question, in what environment were those proteins before they were thrown in the water?
So the life started in the ocean. This is the assumption. And water. Oh, what do you mean? So in a cell. In a cell, so that's more complex. So the proteins come out of the ribosomes and begin. Now the question is.
No. I'm talking about the experiment.
Oh, heat in vitro. That's water. And again, so for many proteins we can, and Anfilsen showed these experiments for the first time, you can unfold them. You unfold them typically by heating, and you can let them fold again. And they do that again and again. In the early days, they showed this because they had some linkers. And then you saw that these linkers were formed or not. Let's not go into the experimental detail. So there are many proteins that you can put into water, just a solution of water, there's a little bit salt in there, and resembles the environment in a cell, and you can fold and unfold the protein by changing the heat. Okay, then you can, at some point you can break it. That's what you do when you boil an egg. The egg gets white because simply the protein becomes bigger when it's unfolded. You essentially, boiling an egg, you unfold proteins. In the egg, they are small, so the protein three-dimensional structure is actually a very small, it's not the smallest possible shape that you can do with this amount of amino acids, but it's very close to the smallest possible shape. Unfolding means making its shape bigger. Unfolding the stretched out chain takes more volume. Occupies more volume simply because you have a lot of binding that brings it close. Binding events, while the stretched out chain doesn't have binding and has a larger volume, the egg is boiled, gets hardened, and you can eat it, or I don't know, maybe you like it raw. There are different shapes. Here are some channels and membranes. Typically in biology what we do, and there was a lot about that last semester, when we start with a sequence here is from peppermint, cytochrome P450, whatever that means, it's a very old protein, let me say that, from peppermint. Then you search the database and you find it, whatever is similar to it. Say you had that and you wouldn't know that it's a peppermint, it's from, what it does. That is the cytochrome P450, and it's this one here. You would just have the sequence. And you wonder what is the function of the thing. Then you find another protein that is similar enough to it, that's alignment technology, or techniques we discussed last semester, sequence alignment. And you find that in spearmint you have this cytochrome P450 that looks very similar. And then you would argue, okay, this one here does the same as that. So cytochrome P450, okay, the plant is a different one here, but essentially you would predict the function of this thing and the structure of this thing by the similarity to that one. Okay? There's the genetic code, so it's triplets that translate to an amino acid. Do I have to get into that for anybody? So on the genome, you essentially, the nucleotides, you have four letters on the DNA level and on the RNA level. There's a slight difference between the DNA and the RNA level, but essentially those are both 4-letter alphabets. Amino acid is a 20-letter alphabet. The way you get from the 4-letter alphabet to the 20-letter alphabet, well, you have to sort of combine the, several elements in the 4-letter alphabet. 4 times 4 is 16 is not enough to make 20 amino acids. Okay, so you need 3. You need to combine 3. Okay, then you have 64, which is more than 20. But what this slide do not get into the detail. Maybe this one here is more, somehow easier to see. This is from Sean and Dangyu. Those are the 4 letters nucleotides, T, C, A, G. Those are all the couplings, the 16 couplings you can do with 4 times 4. And this is the 64, and you see that 64 map to amino acids, and sometimes you have several triplets that map to one amino acid. Sometimes you have only one triplet that maps to an amino acid. Then there are some here used for stop to read and translate into a protein, and that essentially is shown here, and those are the 20 amino acids. Is that enough to essentially, again, the library, the DNA is the library, the protein is the machinery, and you have to translate the information. And that is simply the code, the genetic code. There are about 20,000 proteins in human. They are 35 to 30,000 residues long. There's a length distribution that looks like this. So somehow the average mean is in the ballpark here, but it's a very wide distribution. Let me skip this. This is about the problem why that the bio world, the biosequences are growing faster than the computers, and that puts increasing stress upon computational biology. Another important point for the people who have not visited last semester is proteins come in units, and units that are typically referred to as domains, and you can see them here. So domains historically are things where you say, okay, you have this unit and you see it here again. So if another protein has a similar unit, then you call it a unit. Essentially, it's like a compact 3D thing. So if you just look at this protein, somehow immediately 2 units jump at you. The red and the blue over there. It's just in between. Another way to define it as a physicist, you would say the contacts inside here are higher than the contacts between these 2, and then you have 2 domains. There are different ways of defining them. The important point is they are really built up like Lego bricks, and typically these units do different things. So a unit is specific to a Lego block, and you would see this unit in many other proteins again. Most proteins have more than one Lego block or one domain. And essentially the same sequence typically falls into the same 3D structure. It's not completely the same for function. I'll get to that in a moment. The 20 different amino acids have different properties. Some are positively charged, some are negatively charged, some are polar, here in yellow. Then they're hydrophobic residues, so they want to cling together. Keep water out of them, and the hydrophilic residues bring the water in. Protein structure is essentially, here is one protein, 2 units of a protein in blue and red. Typically easier to visualize them when you put cylinders in. These cylinders are helices. This is a particular ADH, alcohol dehydrogenase, particular protein that consists of 2 works in 2 units. It's called a homodimer. Homodimer essentially means you have the same thing twice and it works together. Now we get to the point of protein function. And that is now where the story gets complicated. So far it was easy. Why is that problematic? Okay, we have the alcohol dehydrogenase. If you had studied chemistry, you would sort of understand what dehydrogenase in principle is. And alcohol you also would sort of understand. In fact, that is the kind of alcohol that you may think about when you think about Oktoberfest. This is the thing that in Asians is not that common. That's why Asians typically have a problem or have a different behavior at the Oktoberfest or at some moments of the Oktoberfest, because simply they lack this enzyme that takes away the alcohol from the blood as efficiently as for most people in this room here or everybody in this room. Now, here we begin to see, in this word here, we begin to see that the story is slightly more complicated. Okay, this is human. That is not that important. But you see that actually there's a particular, it eats a particular alcohol. And what this name here says, in fact, it depends on something that is there in the environment. It's triggered. And that's almost always the case for proteins. They do some function because something makes them do it. Sometimes this is because another protein is close to them. Very often it's another small substrate, glutathione, that is used in order here to trigger some event, opening to be able to process an alcohol. Another aspect of this function here that may not think about, but it's an important one, the two homomers, this is one unit and that's another unit, it's the same protein, just two versions of the same thing. They have to come together. If they don't, it cannot act. This is part of function. Well, you could argue this is part of structure because that's the way it looks in its native shape. But it's not. When you look at the crystal structure, you would essentially see this thing, right? Done over and over again. The binding event here, or the coupling event, the homodimerization that happens, is, if you want, a part of function. And in fact, typically this is the case because the functional site is at the interface of the two. Okay? That's one reason why this can happen. So in some sense, this is all part of the function, but the story gets more complicated. So there is an enzyme nomenclature, and I'll get back into that. It's called enzyme commission. Those things you don't need to learn by heart or anything like that. This is just important. So enzymes catalyze reactions. There are different types of reactions, and there are different ways of catalyzing reactions. Now, if you want to classify enzymes, you would simply write down all the substrates you have, all the types of reactions, and you would come up with numbers that classifies enzymes, right? And that's exactly what this numbering here is about. I'll get into more detail. So ADH is the short for the name of this one here, and it has this number that characterizes this. This one simply says it's about 800 residues long, and catalyzes the fourth step in the metabolism of fructose before glycolysis. And it goes on, and it goes on, and it goes on. The point here is that the explanation of the function is not only the ADH. The explanation of function is also in its environment, in its pathway. What does it do? In which point of the breakdown of alcohol is it actually acting? If you don't have that, you have a problem with alcohol. But it doesn't mean that is the only thing you need in order to digest alcohol. Digestion of alcohol is a long chain of activity. Many, many, many proteins will get active. It's a whole pathway. It's a whole process. And this one here occupies a particular point in that process. This point in itself is also interesting, also important. Some enzymes can act in different points, in different pathways. So that's another aspect of its function, the pathway environment. Okay? So in some sense, what I just wrote in free text is formalized a little bit more here. Here's the chemical reaction. It's oxidation of sugar. So we have much more detail here, but we are still lacking the pathway. The pathway, and now we get into that. KEC is the Kyoto Encyclopedia of... Oh. Tanya, do you know what KEC stands for?
I forgot.
Kyoto Encyclopedia. Okay. Genes and genomes. Thank you. No wonder I didn't know. It has nothing to do with genes and genomes. But so this is one of the central things. The way it happened. This is in Japan, and it started in '92 roundabout. And the idea was, let's just take all the important pathways in a cell and write out every single contributor, every single reaction in an important pathway, one by one. In '92 they started, and now this is by far the most complete encyclopedia that we have. 25 years of 100 people more have been collecting information from the literature, from textbooks. And this is just one example where the ADH, you cannot even see it as part of my slide quality, but it's part of the complexity, is one particular reaction here. This is essentially a cycle that relates to breakdown of sugar of alcohol. And every single of these points here is a reaction. Every one of those, in fact, is a similar story as the one with the ADH. Now, this whole pathway is part of the function, right? Here is a blowout of this, and now we get into a different sort of characterization of function. I said for enzymes, you can essentially write out the substrates, the type of catalysis it does, the type of reaction it does. And that gives you a classification you can put into numbers. Now here's another way. What can we do if the protein is not an enzyme? So can we classify proteins that do other things, that are structural proteins, like membrane proteins? And the idea here was gene ontology. So bring up numbers that would introduce an ontology for all the functions that describe proteins. And they also used numbers for that. And they use a hierarchy. So somehow the higher number, a kinase is something that cuts, is an enzyme that cuts. And what exactly it cuts is the lower point in the hierarchy. The top hierarchy would say it's or the higher in the hierarchy would be the word kinase, it cuts. And then it cuts a bond, it cuts a double bond, and so forth and so on. Now, the problem, well, not problem, the Go started saying that proteins will not only appear in one point of this tree, because we cannot uniquely classify. They're not doing only one thing. There are different ways to look at it. One way is to look at it as they cut. Another way to look at it is they control something. This cutting controls something, and it may appear somewhere else in the hierarchy. And somewhere else in the hierarchy, the level of depth is different than in the kinase level. So if you go 3 levels down in Go, it's not always the same type of information. And proteins are not classified in only one place. They have many domains, and the domains could fall into different regions of the Go tree. Those are, in some sense, when you handle the Go tree, those are problems. It's not a strict hierarchy, and there's redundancy in there. For Michael Ashburner, who started this idea of Go, this was not a negative thing, but it was a power, arguing that this is exactly the way proteins are. They are complex, and Go should reflect that complication. But Go is the attempt to sort of unify biology with a classification scheme that is sort of machine readable or computer oriented, in the sense that it has a hierarchy. It maps the complexity to a hierarchy, but in this map tries not to make the decision when a protein can do 2 things, it can only be one. In the EC, there's a clear assignment, this can only be that. Okay, it cannot appear anywhere else in the EC hierarchy. Go avoided that, because that, in fact, is a problem in the EC. It's good for computer scientists. The hierarchy is completely strict. The first level is always general. The second level is less general. So this is completely going through. That's not the same for Go. The less general part is true. But what is not true is that level 3 is sort of always comparable. For EC it is. But Go is the most comprehensive way of, in fact, defining function, and that would simply be the Go number for that particular ADH. So you can look up the Go number and get these graphs that show part of this metabolic process, the molecular function, the catalytic activity. It is an oxyreductase, and then down here is the aldehyde dehydrogenase activity, which is the alcohol. And you see there are different parts. In our group, so essentially now, protein function is very intuitive, but it's not well defined. You could look at the chemical level, and there you say how the atom is bound has to do with how that protein functions. Or could say that on a biochemical level, it transfers some ion or some atom, a transferase, that is the function of the protein. Or you could say on a cellular level, for instance, it's a kinase. It cuts something and it regulates. Cell cycle control. Or it's developmental. It is set on at some point while the organism is developing, and that is its function. That it starts at that point, time point. And this way, you grow limbs as a fetus or something. Or you could say it has its function as physiological. This one causes a disease. Its function is to cause a disease or not to cause a disease. Or you could say, on a genetic level, its function is essentially whether it's dominant or recessive. So you need only one, and then you knock out the organism or not, where there's a duplicated person. And the problem is all of that is function. Depending on what you work on, you have your preference whether you say that is function of a gene. Geneticists say exactly this sort of level. Chemists would tend to be on, biochemists would tend to be on that level. Our lecture here will mostly stay on sort of this level, biochemical cellular. In our group, we essentially define function as action, so anything that happens to or through a protein, and we try to predict function in some sense. To do that is quite a challenge. Our special angle is evolutionary information and machine learning, and I believe with that we get over to you guys, and we'll continue on that in Thursday in a week from now. Thank you.